Hi, I know it's kind of weird this. Not really done live streaming on YouTube before, okay, but we're just going to kind of see how it goes. Now, what I'm going to do is I'm just going to start off with talking about biology paper 1. And I'm going to kind of go through a number of different biology concepts. Okay, so biology paper 1, I'm going to start off, right, and what I'm going to do is I'm going to talk about the main kind of key questions that we're ever going to need. The first one is the fact we're going to go through these key questions, right, which is define, describe, explain, compare, evaluate, calculate, and then how. Okay, so what I'm looking for is looking for the sort of the key words, right? So firstly, define. All right, so define is a definition. Okay, so if ever you're asked for defining in an exam question, then what you do is you write a definition. So for example, it might say osmosis. What's the definition for osmosis? And then you would say osmosis is the movement of water particles, etc., etc. Okay, so it's kind of key questions that then you need. The next one then is describe. Okay, so then what we've got is we've got describe. Describe is then a big long list of facts. All right, so if I was going to kind of describe my mobile phone, right, then what I would do is I would say it's an orange case and it's probably about an iPhone 2. Okay, and so what it is, it's kind of like a fact base. The next one then is explain. All right, now what you've got to do then is when you're actually doing an explain question, it'll say explain the role of a red blood cell, right? And then what you've got to do in that is you then got to explain, and you've got to say a red blood cell has a biconcave shape. So, okay, so what it is then, it's like a key bit of information, and it says so in the middle of it. So a red blood cell has a biconcave shape, so it can bend through capillaries, okay, because that's kind of the role of it. The next one then is compare, right? So these are then the kind of the key starting words. Compare, you will always put a however in the middle. So for example, if I was then asked about how would you compare an artery and a vein, you would say an artery has high pressure. However, a vein has lower pressure. An artery has a small lumen. However, a vein has a larger lumen. Okay, so if ever you get the compare question, then what you've got to do is you've got to make sure you're chucking a however. Sometimes whereas works better, okay, but compare is still pretty good. Next one is an evaluate. Now, evaluates tend to come right at the kind of the end of an exam paper, all right, and so evaluate, put in a but. So what you'll do is you get a massive, great big paragraph, right, and on that massive, great big paragraph, you've then got to pick out two similar kind of words. And when you've kind of got those two similar words, then what you'll do is you'll say blah, 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 but blah, blah, blah. And it's kind of comparing them together. So it's an evaluate question. Right? Usually worth 6 marks, but you've got to make sure you can kind of say but in the middle. Calculate. Now, calculate is pretty obvious. Right? Calculate means you're going to do a sum. All right. So what you've got to do on the calculate question is, remember, it says calculate. If you're doing the foundation paper, they'll give you the equation, right? But what I'd actually recommend you do anyway is I'd recommend you write out the equation. Okay, so when you write out the equation, then what you can do is you can then write the word convert. And when you write then word convert underneath, if there's anything that does need converting, okay, so remember kind of the little white sheet of all the units that you'll have done hundreds of times, if you then convert to whatever is on that sheet. So what you'll do then is you'll have the equation written. Underneath it, literally write the word convert. So then if you can then write the word convert, and then you'll do, so let's say it's 10 kilometers. Okay, so if it's 10 kilometers, do an equals, and then you will then say 10,000 meters. Once you've done that, once you've done the convert, then what you need to do then is you then need to do substitute. So you've got your equation, you've got an equal sign, and then just literally substitute in the numbers, solve it, and then don't forget to do the unit, right? And I'm going to say it over and over and over and over right? Basically what you need to do is you make sure you need to do every single layer of your working out to be able to get to the correct point. Okay, that's a calculate. Final one is a how question. So how usually means how do you do something? So it could be a method, but make sure you're writing in your equipment. Okay, because if you don't write in your equipment and what you're actually going to do with it, then again, they're going to knock marks off you. All right, so it might be you're doing an electrical one, you say you're going to talk about voltmeters, make sure you write down the fact that you're going to use a voltmeter to calculate or measure the voltage. Or you're going to use a thermometer, and the thermometer there is then to measure the temperature. Okay, so any method, make sure you're chucking in your equipment. Okay, right, now they're the ones. So define is a definition, right? Define, definition, you got loads of definitions to learn. Describe is a list of facts. Explain, make sure you chuck a so in there somewhere, okay, because a so is the word that's then going to give you your extra marks. Compare, make sure you put a however, because if you don't put a however in, then what you're going to do is you're not going to get the comparison word. Evaluate is then a but. Calculate, equation, conversion, substitute, solve, unit, and equipment, or is a how question. Okay, look out for those keywords at the start. Now what I'm going to do is I'm going to go through some of the required practical stuff. Okay, now I'm going to kind of pause for maybe about 20 seconds, right, each time, okay, because no point in pausing for much longer than that, right? And I'm going to go through each one of the biology paper 1 required practicals. So the first one is describe how you prepare a microscope slide. Okay, so that is then the first part of it. Usually onion, but it could be anything. It could be a wasp wing. Describe how we'd use the microscope is then the second part. All right, now in my head, right, what I'm going to do is if I get anything that's kind of method based like that, I will then do a little box in the top right hand corner and I will bullet point it. Okay, and I'm going to say that again as we go down. Okay, and then what you do is you do your plan. That plan, read it back through, and then make sure that that plan then makes sense when you're actually doing your method. Right. Now, 2 methods. Okay, unlikely you're going to get asked how to use a microscope, but you never know. Right, so firstly, right in the middle of the screen there, what I've got is I've got a microscope. Most important bits that you've got on there is you've got your eyepiece. Okay, coarse adjustment, fine adjustment, they're just kind of the dials that are dealing down the side. Right, and then what you've got is you've got your objective lenses, okay, which are the dangly bits. Okay, shortest one is the weakest. And then probably the next most important thing is the stage. Okay, that's where you're actually going to put your slide when it's done. Right, so the method. Now you can kind of elaborate on this a little bit, right? But the method is get a 1 cell thick piece of onion, right? So you cut up your onion, right? And then when it kind of peels open and you can kind of see the skin, get your tweezers, peel off that 1 cell thick piece of onion. I'll try and make sure that it doesn't kind of fold over. Put it on your microscope slide, add 2 to 3 drops of iodine, and what the iodine does, it then stains the cell wall or the cell membranes. Cover the onion with a cover slip, then press gently to get rid of the air bubbles and soak up any excess iodine. And that's as straightforward as it gets, okay, the actual preparing it. Unlikely you're going to get a method of it. Now, I haven't actually seen the paper that you're going to be doing tomorrow, okay, but you never know. Using a microscope slide, right, what you've got to do with that one is you turn the nose piece to select the lowest power objective lens. Okay, so what you're doing now is you're looking at the picture, okay, and the smallest objective lens is the one then that you are going to have pointing downwards. Okay, then when you've got the smallest one pointing downwards, you've got to make sure the gap between the stage and the smallest objective lens is as small as it can possibly be. Okay, now the reason for that is then when you're actually looking down your microscope, the natural thing to do is kind of twist the dial in either direction. But you don't want to do that. What you want to do is you want to twist it in a direction so the objective lens and the stage separate. Because if they go towards each other, you'll break something. So the method on this one is turn the nose piece to select the lowest power objective lens. Okay, usually times 10. And have it as close to the stage as possible. Turn the adjustment knob, the adjustment dial, coarse adjustment, fine adjustment, whichever one you want to talk about, until the cell becomes into focus. Okay, so you're moving the kind of the rough one first. Then, once you've actually got it focused, then what you can then do is you can then have a look at it. You can then have a look at it, and when you can then actually have a look at it, you can then give it a good drawing. Okay, and then when you're having a good drawing, you need to then be able to look down the actual microscope. When you're actually looking down the microscope, you then should have a pencil drawing to be on the right hand side. And if you are ever drawing, make sure you write on the magnifications. Okay, because magnification is important. Okay, and I'm just turning on comments, right? Because somebody then just asked for it. Okay, but they had kind of better be sensible. Okay, right. So what I've then got is that's the method for the microscope. Equation to calculate, right? So once you've actually worked out and given the method, you then need to make sure you remember the AIM equation. AIM or IAM. Okay, it doesn't really matter which way you do it. Okay, and it's the only triangle that I would then probably ever kind of write. The I, and again, what you might get in your exam is you might get a little picture, and that little picture then's got this here, which is called a graticule, which is, I'm never going to be able to write this, I'm doing this with mouse. Graticule, it's a graticule, and what it is, it's like a little, it's like a little ruler within, right? And then what you can do is you can then work out exactly the width of something, okay? And the width is then your image size. All right, if they don't put a ruler, you're gonna have to use a ruler to do it yourself. All right, so what they might do is they might not give you a magnification. They might give you an actual, but they might only give you one number. Right, and if they then do that, what you can then do is you can then use your ruler to work it all out. Next one is, that's then the AIM equation. Highly likely that there's going to be one of those on your exam tomorrow. Highly likely. Okay. Conversions. Right, now, you've all seen these conversions before, right? And the one that you're going to have on the wall in your room is going to have is going to have giga at the top. So remember it: mega, kilo, meter, milli, micro, nano, pico. And also remember, as you're going down, it's always by times 1,000. Okay, it's not times 2,000, it's times by 1,000 each time. And as you're going up the table, okay, as you're going up the table, then it's divided by 1,000. Okay, right, so that's your conversions. And honestly, remember it. The ones that you're likely to get tomorrow is milli, micro, nano. Okay, because it's all going to be about the microscope, and then the other kind of disciplines will then kind of include it all. Right, osmosis. So what you've got to do now is you've now got to think osmosis, and it says define. Okay, so if you're going to do define, then it's a definition. So think about it now in your head, all the different aspects of osmosis and the definitions. Now, I've then just kind of broken it up here. And it says definition of osmosis. Some people like to call it the diffusion of movement of water, okay, because it's kind of like a version of diffusion. Personally, it kind of rolls off my tongue a little bit better saying the movement of water, and it's always water from a high concentration to a low concentration. And again, another kind of little aspect of it could be dilute to concentrated. If you think that when you've got a lot of water in something, so if you've got juice that's very dilute, it's got a lot of water in it. So it's going from dilute to concentrated. Down a concentration gradient. So literally when what you got is you've got a graph like that, high concentration, and then it goes down a concentration gradient. Through a partially permeable membrane without using energy. Okay, and again, it's dead important. Somebody's asking then, can I see the chat? I can, okay, but I'm trying to kind of concentrate on exactly what I'm talking about in the actual lesson itself. All right, so next one, variables. Right, so what we've got now is we've got the variables. This is the phrase. It took me years and years and years right, to basically remember this, and Mr. Tootle told it me, don't mess in class, stay calm. Okay, don't mess in class, stay calm. Dependent is what you measure. All right, now it is, I'm just going to turn the comments off, okay, because it doesn't really seem to be benefiting anybody. Okay, do apologise about that. Put the PowerPoint back on here. Okay, right, so don't mess in class, stay calm. Dependent is what you measure, and always remember it's the first thing you ever measure in any practical. Okay, so whatever you measure, right, I'm not talking about in the osmosis one where you measure the length of it, it's what you're going to measure towards at the end of that individual practical. So it's what you measure. The independent is what you change. So you've done experiment number 1 with your first piece of potato, and at the end of it you've got your dependent variable. Then what are you going to do? Changing it between the first practical and the second practical. That is then your independent variable. It's what you change between practical number 1 and practical number 2. Same is the control, right? And again, a great way of kind of working out control variables is if you look through a method, look for a number, right? And often, whatever that number is, that's often a control variable. Okay, and I'll show you that again in a minute. Okay, right. Now, osmosis required practical. Don't know what they're going to ask. Okay, they could ask anything. They could ask about the equipment, the method, variables, right? But what I'm going to do in a second is I'm going to go through the method itself. Right, so investigate the effect of sugar on a potato. You've got your equipment. If they do ask anything about sort of equipment or things that you're going to use, make sure you list as many things as you can. But unlike what I've done here, write down what they are for. All right, so if they do ask about top hand balance, right, then make sure that you're going to use that to measure, to calculate the weight. A scalpel is then for cutting the potato to the correct length. So if they ask about equipment, tell them what it's for. Now the method itself. Now the method itself is quite difficult to kind of write down. So this is again what I would do is I would then in my top right hand corner I would then do myself a little method, a little plan, and I'd go 1, 2, 3, 4, however many little points you've got. When you've done that little plan, if they do ask for a method, read it back and see if it makes any sense. And see if you think, oh, Mr. Leek's there, and you take the mickey out of me because they're kind of in the wrong order, or I haven't including anything. That little plan can also be used in marking, okay, if the examiner wants to. Now, what I'm going to do now is I'm just going to rub that bit off, okay, but you do yourself a plan if the question is big enough. Part 1, use a cork borer or a core borer to cut 5 potato cylinders of the same diameter. Okay, so it is diameter that you're doing there, which is important. Trim to 30 millimeters, 3 centimeters, you could do 50 millimeters, whatever it is, make sure that you're just writing in the correct number. So they're all the same length. Now the length doesn't matter massively, okay, but it just makes it easier if they're pretty much the same length. Accurately measure the mass of each potato cylinder. Use the word mass, okay, because weight is a force. So measure the mass of each potato cylinder. Now this is again bits that people forget all the time, is what you do is you do your practical, you do your practical, you do your practical, right, but then you don't tell the examiner what you're going to do. Tell them you're going to record it in the table. Right? It might sound dead obvious, right? And you might kind of assume that that's what they're going to think, right? But don't, right? Just don't. Just write down, record your measurements in a table. Then measure 10 centimeter cubed of water. The more numbers you get in, the better it's going to be. And each concentration of sugar or salt solution, and put it in a boiling tube. Again, what you'll notice on that one is you'll notice that they will give you certain concentrations. Add 1 potato cylinder to each boiling tube, leave for 24 hours. Blot them dry with a paper towel, measure the new mass, again it's mass, and record the measurements for each concentration in your table. Okay, so what you've then done, you've done your method, you told the examiner, whoever it is, what you're going to do with everything, and then you've also told them where you're going to write your information. To finish it off, you've got to tell them what you're going to calculate, what you're going to draw, and then what conclusion could be. Calculate the percentage change in mass. Draw a graph. Question what percentage sugar is in the potato. Now I'll run through that one in a second. Right now, if you think about our variables, don't mess. Okay, so the dependent is what we are going to measure. So think about this method here. What are we actually measuring to get a final answer? And the final answer is the percentage change in mass. Okay, that is what we're trying to work out, which is our dependent. Don't mess. In class, what are we going to change? Measure and each concentration of sugar solution. So what it actually tells you there is it tells you what you're going to change. So you're going to change the concentration of sugar or salt, depending on what they ask. Stay calm is then our control variables. Now, like I said before, right, if you do a method, look for numbers. Right now, I know diameter isn't a number, right, but the reason why you use a core borer is to get the same diameter. So therefore, that's a control variable. Trim to 30 millimeters. There's a number, and it's a control variable. That's what stays the same. 10 centimeter cubed of liquid. Okay, so 10 centimeter cubed of liquid, that's a control variable. 24 hours is a control variable. Right, so numbers that are in there are often the best ones that you're going to use. Okay, right, then that's a diagram. So that diagram gives you a little bit of an idea about what's going on. The final question then is, right, you've got the variables, you've got your percentage change in mass. Okay, you then get a graph that looks like this in the bottom right hand corner. Okay, so that then is the, oh sorry, I haven't done that, right? So then what you've got then is you've got a graph that is in this bottom right hand corner here. So the graph is the percentage change in mass at the side, and what you can do is you can see it starts at 0. Okay, it goes up, which means it gets bigger, so it gets bigger that way, and it gets smaller that way. Along here is the concentrations. Now the part that they're going to ask for right at the end is where that red circle is there. That red circle tells you the amount of sugar in that individual potato. So the question could be, I think I had it previously, what percentage of sugar is in the potato? You look at where your line curves and crosses that axis, and wherever it crosses that axis there is the concentration of sugar in the potato. That'll be your final mark. Right, here's another question. Investigate the effect of salt in a turnip. Okay, doesn't matter what vegetable it is. When the last time Osmosis was on, or one of the last times, it was all about carrot. Everybody comes out of the exam saying, oh, you didn't tell us about carrots. Right, doesn't matter. Right, it's all the same. All they're trying to do is just catch you out. Right, food composition of a crisp. So what I'm going to do is I'm going to speed up a little bit here. Okay, investigate the food composition of a crisp. So you've got 4 food tests. You've got your fats, you've got your proteins, you've got your carbohydrates, and you've got your starch. Okay, they are then the 4 food tests. All right. Now, whenever it says investigate the food composition of a crisp, even though it might not be worth any marks, but it could be, I will always go for my preparing a food sample first. Now, the reason why that's always good is because if you then start talking about the food sample, right, in your method itself, they might be expecting you to have said what the food sample is. Okay, and it's also good practice, because if you think about it, if you've got a burger, right, big brown burger, and then what you do is you just shove it into a boiling tube, and then you add some of the solutions to it, do whatever you've got to do, and all it'll do is just look like a big brown sludge. So if you're asked about food tests, and it's a decent sized question, make sure you do preparing a food sample first. Put the water, or put some of the food sample into a mortar and pestle with some distilled water. Filter it. And then when you get your filtrate out, the filtrate is then lovely and clear, and then what you can do most of your food tests with it. All right, so big question on food tests, not just a one-liner, right? Talk about preparing your food sample. Starch test, iodine, right? And what it does is it starts off orangey brown. I should have put that on there really. So it starts off orangey brown, okay, which is kind of the expected color. Add a few drops of iodine. If it turns, or if there's starch present, it goes blue-black. Okay, and it's often done, this one, in a spotting tile. Okay, and it's also one potentially that you could do without doing the actual preparation, because you can put crisps and bread and things like that in there. Okay, so iodine test for starch. Starts off orangey brown. Positive result is blue-black. Don't forget to write down both colours. Benedict's test, most likely one you're ever going to get. Okay, so the Benedict's test is a test for sugar. Okay, put your food sample in a test tube, add a few drops. Let's not go for few, let's go for 5 drops of Benedict solution. Better to be more accurate. In the sample on the test tube, okay, put the test tube in a water bath. Okay, so this could be a 3-mark question, right, where what you've got to do is you've got to name the test. You're then going to make sure that you mention the fact that you put it in a water bath. Don't put it under a Bunsen burner, because a Bunsen burner will just incinerate it straight away. 80 degrees C for 5 minutes. Tricky numbers. The colouring starts off blue. Okay, it's got copper sulfate blue. A lot is red. Right, and again, people taught me this probably about 5 years ago. Rainbow backwards. Red, orange, yellow, green, blue. Forget about indigo and violet. So a lot of sugar, and this is the rainbow backwards. Benedict's test. Don't forget, 80 degrees C in a water bath, not a Bunsen burner, a water bath. Biuret test. Okay, food sample in a test tube, 1 centimetre biuretic solution A and 1 centimetre cubed of biuret solution B into the test tube. Sometimes they might just put the liquids together, sometimes they might not. Now the way this works is biuret solution is copper sulfate again, okay, and then the other part of it is then an alkali. When you put them in there, shake the test tube gently, any colour changes. So it starts off blue, that means there's no proteins, and it goes purple, which then means there is proteins present. Final one, there's 3 tests then for lipids. Number 1 is a few drops of sample in the test tube, a few drops of distilled water and a few drops of ethanol. Okay, it isn't just a test with ethanol, it's a test of ethanol and water. So you've got to mention both of them. Shake them, see what you see. If it goes milky white from a clear liquid, I guess the clear liquid is literally a bit like vodka really, it's just water and ethanol. If it goes milky white and it tends to kind of appear at the top, that means there's ethanol present. Okay. Second one is get a piece of brown paper, like brown paper in the kitchen, and then what you do is you just get your thumb, rub your thumb with the food underneath. This is then probably the one where you would just use the food again, just like with the starch test. Let it dry, scrape off any little chunks of food, and then if it is clear, the paper like that, that means there's fat present. If it isn't, then that means there isn't any fat present. Okay, the third one is Sudan 3. Now, Sudan 3 isn't kind of highlighted on the syllabus, right? But an examiner friend that I kind of know said it might. Okay, starts off a clear transparent liquid. That's Sudan 3, and then what it does, it produces a beautiful red. It's kind of like a vivid red orange solution. Kind of looks really good. Right, enzymes. What conditions affect the activity of enzymes, and what happens if the conditions are wrong? Right, there's 2 graphs. Could get a graph, could interpret them. Right, so the one on the left here is the temperature one, and the one on the right is then pH. Now, what I'm going to do is I'm actually going to start with the one on the right first. Now, what this is, it's enzyme activity with pH. What you can do is you can see the peak is approximately, let's change colour, let's go for green. The peak is approximately pH 7, okay, which is pretty much what your body is. On either side, if the pH increases, Okay, so when I say increases, that means the number's getting bigger. What happens is it drops, and that's your denature. Okay, so don't forget denature. Right, what denature is, that's then where the active site of the enzyme changes shape, and then this substrate then can't connect in and have any sort of chemical reaction with the enzyme. If it gets lower, more acidic, it also denatures. Okay, again, what's happening is the shape of the active site is changing due to an increase in pH or a decrease in pH. Now, another one they might do is they might chuck in a graph there. All right, and they might say, what's that? Right? Why isn't it all denatured? And what you've got to do is you've got to remember an enzyme that is pretty much at pH 3 or 2, which is then your pepsin. Okay, pepsin in your stomach is kind of the second graph that they might do, because most of your body is obviously pH 7. Now, temperature is slightly different. Okay, it's slightly different in the fact that your optimum temperature is about 37 degrees. Okay, so every single one of you sat there now is about 37 degrees. If it goes warmer, what happens? It denatures. Okay, and it denatures quite rapidly. So the enzyme's active site is then changing. Now, cooling down, it doesn't denature. What happens is there's less kinetic energy, there's less movement, so there is less chance of the enzyme and the substrate meeting. So the activity decreases. So on the left-hand side of this one, there now isn't any denature that's going on. Okay, that's enzymes. Right, now that's kind of summarizing exactly what I then just said. Right, so if the temperature is too high, it denatures. If the pH is too high or too low, it denatures. And that is then the changing of the shape of the active site. Right, how do enzymes work? Quick one. Now, lock and key model is the way that an enzyme works. It starts off enzyme and the substrate goes in. And what it does, it fits in perfectly. The enzyme substrate complex, so this is then stage number 2. The enzyme does its job, breaks it up, or it can build proteins. Out they come, and then what you do is you get 2 products. The enzyme then repeats. Okay, so then what it does is it just keeps on going over and over and over again. All right, now this is again quite a good definition. This can be definition of digestion, or it can be the definition of enzymes. So digestion or digestive enzymes, they speed up the conversion of large insoluble molecules, right? So like your burger, like your chips, whatever it may well be, into small soluble molecules that can be absorbed into the bloodstream. Okay, that's a good definition. That's what digestion is, right? And it's also how a digestive enzyme works. Main digestive enzymes, and where do we find them? Okay, there's a little table. So the 3 enzymes, we've got carbohydrates, often called amylase, and we know it's an enzyme because it's got ASE on the end. We've got protease, ASE on the end, breaks down proteins. Lipase breaks down lipids, and bile is then kind of like the 4th one. It's not an enzyme, but we'll talk about it anyway. So carbohydrates. Think about your digestive system. Food goes in your mouth, and the first enzyme that hits it is amylase. So therefore, it is made in the salivary glands. So carbohydrates start acting in the salivary glands. Then it's pretty standard. The others, you've got pancreas, and the small intestine also produce carbohydrates. And remember, carbohydrates, they break down into glucose, which is there. Okay, carbohydrates, amylase breaks down starches into glucose. Protease, the first dealing with proteases is the stomach. Pepsin, already talked about it. They're also made in the pancreas. Okay, break down proteins into amino acids. So basically what a protein is, it's a chain, loads of amino acids all in one big long line, and the protease comes along and goes chop, 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 and they're all chopped up. Lipases, made in the pancreas, work in the small intestine. Right, so you can't remember about enzymes, just write pancreas. they break down lipids into glycerol and fatty acids. I don't know why, but fatty acids and glycerol always kind of springs off my tongue a little bit better. Now, the last one isn't an enzyme, but it has a pretty significant kind of role in the digestive system. It's made in the liver, stored in the gallbladder. And what it does is it emulsifies lipids. Okay, so after this, if you then go in the kitchen and you have a bacon butty, right, what you got is you got the bacon butty and there's loads of fat on it. Right, that goes in the sink, and then you can see all the globules of fat on the top of the sink. Put in your fairy liquid, and the fairy liquid emulsifies the fats and it breaks them down. And the idea of that is you get a larger surface area for the other or the lipases to then work on. Okay, so they're the enzymes, and it's pretty important that you know exactly where they are and where they start. Bile emulsifies fats in the digestive system, and it also has another role, which is then neutralizes the stomach acid pH. Right, required practicals again. Here's another one. Investigate the effect of amylase in different temperatures. Okay, got this one. Right, I'm asked to write a method. So what I'm going to do is I'm going to write my method in the top corner. When I've written my method in the top corner, okay, as like a little plan, I'm then going to work through it. Now, there's a key point in this one that I'm going to highlight in a second. So what you do, heat your water bath 35 degrees C, 2 centimetre cubed of buffer solution. So what the buffered solution means is it keeps it at a certain pH. So it's a buffer pH 7, it will stay at pH 7. If it's a buffer pH 6, it will stay at pH 6. 4 centimetre cubes of starch solution. So what you're basically going to have, you can basically have 3 boiling tubes, one with buffer, one with starch, and one with amylase. Now there's more amylase in there because you're going to use 5 lots of 2. Allow them all to reach 35 degrees C. Put 1 drop of iodine solution into each depression on your spotting tile, which is then there. Use a pipette, 2 centimetre of amylase solution to the mixture, start the stop clock. So basically then what you're doing is you're going to get some amylase and you're going to mix it with the starch and the buffer solution. When it first starts, what you're going to have is you're going to have your spotting tile, and the first one is just going to go black, right? Because what you've got is you've got the starch still in there. Okay, so it's going to go black. The next one will probably go black. The next one after that will probably go black. And the next one after that will probably go, or could go, orangey brown. Right, now if that goes orangey brown, something has then happened. Okay, and this is then the key part to it. The key part to this now is that the amylase has then broken down the starch and turned it into glucose. So the amylase enzyme has broken down the starch, so now the starch isn't present, so now this now won't go black because the starch is gone. You keep doing that for 30 seconds, use a glass rod to remove 1 drop of the mixture, keep sampling every 30 seconds until the iodine doesn't change colour. Then you record it on a table. Then you repeat it, and when you repeat it, you can then do it at a different temperature, because the initial question was investigate the effect of amylase in different temperatures. Okay, right, so there's your spotting tile. Those are the 2 graphs that we talked about before. Don't mess in class. So the dependent variable is what we measure. Okay, so we're not measuring the quantities of the liquid. What we're measuring is we're measuring the time for the iodine to stop changing colour. Don't mess in class, right? What are we changing? Now for this one, that's not right. Okay, we're changing the temperature. Control variables is the pH, is the iodine concentration, the volume of the solutions, and again, look for any words that you might, or numbers rather, that you might have. Right? This then shows kind of a bit of an idea about what the colours are. Okay, so if it's quick, so we're talking about 30 degrees C or 40 degrees C is even quicker. Starts off black, then what it does, it goes brown quick. So the quicker it stops changing colour, the better the temperature is. Right. The alternative, that then I guess some of those pictures then said, is amylase at different pHs. They could also ask different enzymes. It doesn't really matter what enzyme they talk about. Okay, just go along with whichever one they say. Here's a quick skills thing, peer review. Right, so peer review is then when you do a test and you've got to get someone to check on it. Main reasons for it: detect false claims. So somebody that then comes up with this fantastic idea, but it's a load of rubbish. Inaccurate data mistakes. Okay, so anybody that's then checking things, if something's inaccurate, then somebody else is checking it. This is why peer review. Removes bias. Like, so if I was then inventing a drug, right, then what I'll probably do is I'll probably make it as biased as I possibly can to get it through, so that then I can make lots of money. So peer review gets rid of bias. It also verifies new data, and it provides a consensus, right? So when there's kind of big decisions that are made, lots of scientists then need to be involved. That's peer review. Okay. Now, a little bit of kind of factual stuff. Carbon dioxide in the leaf. There's a leaf, right? I was going to go for green then, but green probably won't show it very well, so I'm going to do a bit of purple. Okay. So what I've now got is my leaf. Stoma allows air to enter. Right. So let's start at the top. We've got the waxy cuticle. Now the job of the waxy cuticle is to keep water out. To keep water in, right? So if you think about a cactus has a thick waxy layer to stop water getting out. So it's not a waterproof layer, it's to stop water getting out. Upper epidermis, palisade cells, okay, that are rectangular, xylem and phloem, spongy mesophyll that's got gaps in between, lower epidermis, stoma and guard cell. So how does oxygen get in? It goes through the stoma, through the air spaces, and it gets to the palisade cells that tend to have the most chlorophyll. Once the photosynthesis process has then happened, the carbon dioxide comes out of the palisade cells, through the little air spaces, and out of the guard cell. Right, what that does is the guard cells control the opening and closing of the stomata, because basically what the stomata is, it's just a little hole. All right. Xylem and phloem, okay, favourites. Now these again, two of the words that you must spell. So the adaptions of xylem is it carries water and minerals. One way, which is up. They are dead, and they also have no end walls. So these kind of little gaps that are here, right, it's where all the cells have died. So it's literally like a big straw that goes all the way up a tree. Phloem, on the other hand, okay, is carries organic molecules. So often it would be kind of glucose and anything else like your proteins, any other substances that the tree might need somewhere, and it goes anywhere. So basically you can go from a leaf to a root, you can go from a root to a leaf, you can go trunk to leaf, wherever it wants to go. All the cells are living, okay, but kind of one of the key adaptations is the fact that they have end plates. And these are the end plates, and what they are is they're kind of the ends of the cells are called sieve plates, and what they have is they have little holes, and those little holes will then decide where each individual organic molecule is going to go. Now, this is a biggie, right? It's kind of one that doesn't come up very much, but when it does come up, it's an absolute steal if you can remember it. So, how do plants produce glucose, and what do they use them for? So the glucose is produced in photosynthesis. You've got to write that whole equation. 6 CO2, don't forget the 6. So that's the gas that's taken in. You've then got your water, again 6. Light is the energy source, and that then tells you it's also an endothermic reaction because it's taking energy in. Glucose, C6H12O6, and oxygen, 6O2. Okay, photosynthesis. So that is then where the glucose comes from in the leaves. Now, what is it used for? Right, this is a great 6-mark, 4-mark question. Glucose in a plant is used for respiration. Okay, because don't forget that plants don't just photosynthesize, they are continuously respiring as well. So they're doing exactly the same as us, right? But people forget that they're actually respiring because they are growing as well. It can be stored as insoluble starch. Think about a potato. So the glucose goes all the way down the plant to the potato, and then what it does, it gets turned into starch. Stored as fats and oils, nuts, okay, made into cellulose for cell walls, and it also combines with nitrates to make proteins. Right, so they are 5 great uses of glucose. What conditions affect it? Temperature, light intensity, and carbon dioxide concentration. Okay, so they are the 3 kind of main conditions that affect the rate of photosynthesis. And it kind of makes sense, doesn't it, really? If you look at the graph, rate of photosynthesis, when it's cold, 0 degrees C, it's slow because the particles don't have very much kinetic energy. When they gain more energy, when it gets warmer, the rate increases. And then when it gets too hot, the rate decreases. Okay, because often that can then be where the proteins and the enzymes are starting to denature. Light intensity, if there's no light, there isn't going to be any reaction at all. As the light increases, so light intensity increases, right, it gets to a certain level, and at that certain level, it then tails off, because that is then the limiting factor, right, the fact that the plants literally can't use the light that's there. Carbon dioxide concentration follows very similar, where it goes up and up and up until it gets to X, right, which is then the maximum, and that is then your limiting factor again. The final one that you could use is the amount of chlorophyll, right? So if you haven't got very much chlorophyll, you're then going to not do much photosynthesis. So things like variegated plants, so a variegated plant where it's got green, right, and it's got yellow bits there as well. Okay, so it's going to have less chlorophyll for the photosynthesis. Okay, this is the last required practical. Absolute rubbish, right? I don't know how anybody could possibly think that this is any good. Right, describe how we can measure the rate of photosynthesis using pondweed. Right, so you'll all have come across it. These are the 2 setups. We've got a lamp, we've got a ruler, and then we've got a boiling tube with pondweed in it. You could also have a light source, and then this time what you've got a beaker, okay, and that beaker has then got water in it. The pondweed, then what it does its photosynthesis, and the idea then is you are going to collect the oxygen at the top. And it could say, how do you know it's oxygen? Relight glowing splint. Now I've done this probably 400 times, right? I don't think I've ever collected more than about 2 bubbles. Okay, so these are then the 2 possible setups. Right, so method. 10 centimeter piece of pondweed, cut edge at the top. Okay, so now the idea of that then is you will then put the pondweed into your boiling tube or wherever it is, you get your scissors and you cut it underneath the actual water itself. Because the idea then is you're then going to not allow any air bubbles to go inside the stem. You're going to add sodium hydrogen carbonate. Now, sodium hydrogen carbonate is it's a substance that provides carbon dioxide. So it kind of encourages the growth. Right, so now what it is, it's in the liquid. Fill the measuring cylinder with water and sodium hydrogen carbonate and put it over the weed. Position the beaker of pondweed 1 meter away from the light source. So then what you've got is you've got your pondweed in whichever setup you've got, and it is 1 meter away. Okay, start far away. Once it's then 1 meter away, start your stop clock and count and record the number of bubbles released in 3 minutes or the amount of gas produced. Okay, very exciting. You probably run out of fingers, right? Maybe not. Collected in the measuring cylinder in the same 3 minutes. Record your results on a table. See that? Record your results on a table. Move the light source so that the pond is 80 centimeters away, but must allow acclimatization. And I would say 5 minutes. Repeat 60, 40, 20. Calculate the rate of bubbles produced. Calculate the light intensity. Okay, so what I'm going to do in a second is I'm just going to talk about the variables and calculations. Okay, so variables, don't mess, right? So dependent, what I measure, number of bubbles, and the volume of gas, right? Make sure you're using the right terminology. Don't mess in class. Independent is what you change, is the distance the light is from the pondweed. Control, I'm just going to go back to the method. Let's have a look. Control is 10 centimetre piece of pondweed. Okay, you're going to use the same concentration of sodium hydrogen carbonate. You're going to count and record the bubbles for 3 minutes. So what I'm doing here is I'm just literally scanning for numbers. Acclimatize for 5 minutes. Standard, it goes 80, 60, 40, 20. The gap between each one is 20 centimeters. Right, so your control variables look for numbers. So time to acclimatize, concentration, length of the pondweed, any numbers that you've got. All right, calculating light intensity is 1 over distance squared, and the distance squared needs to be in centimeters. And an approximation of graph that you might get would be rate of photosynthesis against light intensity. So what you can do is you can see as the light intensity increases, the rate of photosynthesis increases until it gets to a certain point where it becomes the limiting factor. Okay, right, a little bit more factual stuff, and I've literally got probably about 8 more kind of slides to go through. Okay, gas exchange or exchange surfaces. Again, this is a dead popular question, right? And this is again one that people got caught out a number of years ago because they said explain the role of an exchange surface in the gills of a fish, right? And nobody could remember the gills of the fish one. So there is 3 answers, and all you've got to do is if you see the words exchange surface in your exam, you're going to write these 3 down. Now, the most popular ones are the villi in the small intestine. You've got the alveoli in your lungs, and I guess you've got the gills of the fish as well. So, exchange surface. It will have a good blood supply. Alveoli, villi, gills of fish have all got good blood supply. You will be asked to explain. So you will then say, so it can maintain a concentration gradient. The villi also have a thin wall, often 1 cell thick, so there is a short diffusion distance. The alveoli, villi, gills of a fish have a large surface area. This is due to the villi, the alveoli, or the gills of a fish to increase diffusion. Okay, so it could give it a short diffusion pathway. And that will then be a so. Oh, I didn't say a so then, okay, but it should be a so in there. Right, they're typical examples. You've got your small intestine is your villi, you've got your lungs, your alveoli, gills of a fish. That also happens in plants as well, root hair and leaves. Now, just an example of a comparison question, kind of going right the way back to the start where we talked about that. So compare aerobic and anaerobic respiration. Now what I've done here is I've just done aerobic first. Now remember, if you're doing a comparison question, you're going to chuck in a however. I'll finish off. However. Okay, so aerobic respiration is where there's enough oxygen and there's a lot of energy produced, and it's done in the mitochondria. Okay, so what happens is glucose is oxidized. Aerobic respiration releases large amounts of energy. Right, so what I'm doing here is I'm just introducing the facts about aerobic respiration, and here's the formula. C6H12O6 plus oxygen, should really put 6 in there, gives 6 CO2 plus 6 H2O. Right, they're facts about aerobic respiration. Anaerobic respiration. Anaerobic releases a much smaller amount of energy, and it is the incomplete oxidation of glucose. And basically what happens this time is the glucose, it doesn't turn into energy entirely, it turns into lactic acid and then just gives out a small amount of energy. Now the comparison question is, compare aerobic and anaerobic respiration. You will then say, in aerobic respiration, glucose is oxidized by oxygen to release energy. However, in anaerobic respiration, the oxidation of glucose is incomplete, causing a buildup of lactic acid. Also, aerobic respiration produces large amounts of energy, and in anaerobic respiration, much less energy is then produced. Equations, they might ask them, so what I've done is just put them in. But don't forget, I'm not sure if I actually did that then. Okay, chuck in a however. Then the other one that they might talk about is anaerobic respiration in yeast. Okay, so what happens this time is in yeast, you've got glucose, which is C6H12O6. That turns into ethanol and carbon dioxide. So again, in anaerobic respiration, where in humans, it's not ethanol, it's lactic acid, and you get carbon dioxide. Quick definition question, define metabolism and list 3 of the main reactants. Metabolism is the sum of all the chemical reactions that take place within each cell of a living organism that provide the energy. Okay, so metabolism is the sum of all the chemical reactions that happen within a cell in an organism. A few of the reactions could be conversion of glucose to starch, it could be making lipids, it could be using the glucose, it could be respiration, and it could be the breaking down of proteins to form urea in excretion. Right, so what you've got to do then is just got to think of any reactants or reactions that might happen. So respiration is a brilliant one, because you all know that one. Urea is then where you've got proteins that are then broken down, and converting glucose to starch in a plant. Now, I've basically come to the end of what I'm then going to be talking about. Now, the final kind of tip really is, whatever the question says in front of you, right, read it and totally 100% understand exactly what it's asking you. So read it, then read it again, and then think, what is it asking you? And look at every single word in that question, and every single word in that question is giving you a clue what to write. And then just remember all those kind of key sort of starting words that I had right at the very start. These words here also tell you what you've got to write. Okay, but I'll just say it again. Look at the question, and every single little word in that question tells you what you are going to be writing in your answer. Okay, analyze it, pause, take your time. Right, you've got an hour and a quarter, you've got an hour and a half, or hour and 3/4 rather. Make sure you know exactly what's then being asked. Right. I'm going to go now. It's 7 o'clock, time for tea. Okay, and good luck tomorrow. Take your time, read the question. That's the main tip that I'm ever going to ask. I will see some of you tomorrow.